today we'll discuss about sequencing. What is sequencing? When you have a DNA, for example, you have a double-stranded DNA. The bases which is being present is being unknown. So you want to know the order of the bases. So in order to know the order of the bases, you should go for the sequencing. So if you check the methods of sequencing, the first method which has been introduced is Maxam-Gilbert's method. So what is Maxam-Gilbert's method? Maxam-Gilbert's method is a method where few chemicals is being used for the conversion of the bases or for the cleavage of the bases. So once the Maxam-Gilbert method is being adapted, you have to take the double-stranded DNA, label it. After labeling, you have to cut out. So the cut out fragments is being divided into 4 portions. So each tube containing 4 fragments. For example, you will be having a fragment of G, A plus G, C and C plus T fragments. So these are being taken in 4 different tubes and the chemicals is being added. Once this has been done, the cleavage of particular bases will occur. Once the bases is being cleaved, you can put into the agarose gel and find out what is the pattern of arrangement, what is the array of bases which is being present. But this is the oldest technique. Only you can go for sequencing only 500 bases. But there are method which gives more than 500 bases to be sequenced at a time. The second method which has been generated after Maxam-Gilbert method to improve the efficiency is your Sanger method. The Sanger method is still not followed widely. So what is meant by Sanger method of your DNA sequencing? So Sanger method of DNA sequencing uses PCR as the basic principle. So if it uses PCR as the basic principle, we should have a template DNA. For this template DNA, we need a primer. So a primer to be attached to a template DNA. So when you put the primer and give proper bases, the tag polymerase will go for the extension. But when in case of your Sanger's dideoxy method, you should have 2 DNTPs. One is your DNTP and other one is your DDNTP. So what is your difference between DNTP and DDNTP? So your DNTP will be having 3' hydroxy and DDNTP will be having 3' H. That means the hydroxyl group from your 3' end is being removed. So the further bases cannot be added. So how to carry out the Sanger's dideoxy method? You have to take the template. The template should be put into 4 different tubes. So after that you should add 4 different bases. That is your DNTPs, that is DDATP, DDCTP, DDGTP and DDCTP. All are put in 4 different tubes. Then you should add your DDNTPs, that is DDATP, DDTTP, DDCTP and DDGTP in corresponding tubes. Once when you take this DNTP, you should take in the ratio of 1:100. That means your DDNTP should be 1 time and your DNTP should be 100 times. Once this has been added, what will happen? In the denaturation, the strands will get separated. Once the strands will get separated, your primer will go and attach. Once the primer is being attached, you will have the reaction forward. That means the synthesis of DNA from the 5' reaction towards your 3'. So once when the bases is being added, you will have a complete strand of your DNA. So if any DDNTP is being added, what happens exactly? You will be having the addition of bases which is being stopped because your 3' end is being blocked. There is no OH to form phosphodiester bond with the phosphate which will be present at the other base. That means you will be having winding of the reaction. The terminator sequence, that is DDNTP, if it is being added, your chain reaction terminates. So you can call Sanger's dideoxy method as chain termination method. So in chain termination method, there will not be any further extension. So this will be done in 4 different tubes, that is in your DDATP tube, DDGTP tube, DDCTP tube and DDTTP tube. So once these tubes has been synthesis, you have to load each tube in 4 different wells of your agarose gel electrophoresis. Once when you load it, you will be able to find out the amplified bands. So the amplified bands which contains the N sequence as A will be given the band A. If it contains G, then it will be given as G. Similarly, you can analyze from the bottom towards the top, that is from your 5' end towards your 3' end, you can get the array of your bases. So this is your Sanger's dideoxy method. Suppose if you have a genomic DNA, this genomic DNA should be sequenced soon, that means very shortly, then you can go for one of the recent method which is available. It's nothing but your genome shotgun method of sequencing. The entire genome should be taken and it should be fragmented with restriction enzyme. So once the genomic DNA is being treated with your restriction enzyme, you will be able to get the fragments. You will be having different fragments which are of shorter size. So these fragments should be further cloned. When you go for cloning, this should be put into a suitable vector. Once you obtain the short fragments of DNA in shotgun method of sequencing, the short fragments should be put into the suitable vectors. So you have the vector with the short fragment which is being entered. Like this, you will be having numerous fragments which has been inserted into the vector. After this, you should go for the cantic formation. So the cantic will be formed from the clones which has been formed. So how to form a cantic? The overlap of your first sequence, that means the first sequence will overlap with the second and second with the third. The first sequence or end sequence is the start sequence of the second, and second sequence end sequence is the start sequence of the third. So if you go for this construction, you will be able to get the entire chromosomal DNA. So the genomic DNA which has been fragmented out using your restriction enzyme can be obtained in the form of your array. The array will give you the set of bases will be present. So this is one of the technology where you can go for the shotgun method of sequencing to get the entire genomic DNA to be sequenced. Other than this, we have other methods of your next generation sequencing, and the next generation sequencing mainly uses the principle of your PCR as well as the treatment with other components also. So we'll have the NGS later.